Hello everybody, this is Alex here, and today I'll be giving you a brief presentation of the Golgi apparatus and lysosomes. So without further ado, let's get started. This is a picture of Camillo Golgi. Camillo Golgi found the Golgi apparatus in the year 1898, and here is a sample of the organelle that he found. The Golgi apparatus can be seen under a light microscope. And here are some samples of the organelle being viewed under an electron microscope. The Golgi apparatus consists stacks of flattened membrane-bound sacs called the cisternae. And these organelle, they are found in all plant cells and animal cells. The number of Golgi body actually differs, as in animal cells they tend to be fewer and larger, and in plant cells they are smaller but they consist a lot. Stacks of sacs and Golgi vesicles will then form the Golgi apparatus. This circle here is known as the transport vesicle, and the rest of the body here is called the stack of sacs. The Golgi apparatus is located near the endoplasmic reticulum. To be more specific, it's the rough endoplasmic reticulum. As we can see here, the rough endoplasmic reticulum, which is located right beside the Golgi apparatus, will produce transport vesicles which will then be transported to the Golgi apparatus. The Golgi apparatus has two phases, the cis phase and the trans phase. The cis phase would be the convex shaped part of the Golgi apparatus, whereas the concave shaped part is the trans phase. The vesicles will then move from the cis phase to the trans phase at that particular order. The Golgi apparatus receives and chemically modifies two products, which are proteins and lipids. And here are some modification examples. Addition of carbohydrates, proteins to form glycoproteins, and adding or deleting specific sugar molecules to modify the branch sugar structure found on proteins. The modified proteins and lipids are then transported into Golgi vesicles, and they are sent to three locations. And these locations are within the cell, to the plasma membrane, and outside of the cell, depending on what substance. Within the cell, it's sent to the lysosome. Primarily, enzymes will be sent to the lysosome, which contain protease. Whereas in the plasma membrane, vesicles fuse with the cell membranes to form carrier and channel proteins in the phospholipid bilayer. The Golgi vesicles secreted by the Golgi body will be released out to the cell. This process is called exocytosis. And right now I'm going to give you some information about the lysosome. And so here we go. The lysosome can be seen under an electron microscope, as the picture shown here. It is 0.1 to 0.5 micrometers in diameter, and the functions. They produce hydrolytic enzymes, lysosomes, and digestive enzymes, which break down unwanted structures and here are some hydrolytic enzymes. Some other functions of the lysosome would be autophagy. Autolysis, self-digestion of a cell through the release of acetic lysosome within the cell. For example, would be metamorphosis of the tadpole tail. And finally, the phagocytosis, which digests bacteria by endocytosis. This process is also called cell eating.